Welcome to your lab manual. What we're looking at in this video is the introduction to the lab manual. And there's a number of things we need to know from the introduction before we actually get to lab 1. Now, the first lab period or lab meeting we have during the semester, we use to introduce the laboratory. The second lab meeting of the semester we use to do safety training, mandatory safety training. And the third lab meeting is when we actually do lab 1. So the third scheduled meeting for lab is when we'll actually do our first lab, lab 1. But there are things in the introduction that you'll need to know for that. Now let me remind you that at the end of every lab exercise, there are a list of performance objectives that tell you what you need to know for the lab quizzes in the laboratory portion of the course. And there are several categories of objectives. Many of them begin with verbs like define, state, discuss, describe, or differentiate. And when these are used, you would be expected then to be able to do that or answer that by means of a short answer, multiple choice, or matching question on our lab quizzes. So we do cover a fair amount of content in our lab. When verbs like demonstrate or perform are used, this is where you'd be expected to demonstrate a particular technique or procedure during the course of the lab. And when we have traditional labs, those are what we call core lab points, where you demonstrate certain techniques or things you found to me. When verbs like recognize or interpret are used, then you're expected to be able to answer that by means of a practical question. So each lab quiz will have theoretical questions covering the content of the lab, and they will have practical questions. And then the performance objectives are the ones that you'd be graded for by means of your core lab points. And all of that will be explained in your syllabus, in your Getting Started course tour, and in our first lab meeting. So to start out, the introduction does have performance objectives at the end. If we scroll down to the end of the lab, you'll see the performance objectives for the introduction. So the first lab quiz we have during the semester covers the introduction to the lab manual, this right here, as well as labs 1 through 4. Now you notice as you go through the objectives, some of these are non-testable in terms of writing on an exam, like follow the laboratory rules. But again, if you're not following laboratory rules, you could lose lab core points for performance. But some of these are definitely testable. For example, under binomial nomenclature, it says you should define genus and species and correctly write the scientific name of a microorganism. Under metric length, it says define and give the commonly used abbreviations for the following units of metric length and fluid volume: centimeter, millimeter, micrometer, nanometer, milliliter, and microliter, the units of measure that we'll be using during the course. Likewise, state the length and width of an average rod-shaped bacterium and the diameter of an average coccus-shaped bacterium. There are a number of objectives under using the microscope. Many of them say you should be able to use the microscope, which again would be core lab points. You'll be finding stuff and showing it to me during the labs where we use microscopes. But there are a few testable ones here as well, like define ocular lens and objective lens, and state the reason for using immersion oil at 1,000 magnification, and calculate the total magnification of lens system when using a 10x, 40x, or 100x objective in conjunction with a 10x eyepiece. So keep in mind there are some testable objectives here that you will see on the first lab quiz. And also keep in mind, after the performance objectives, each online lab has a self-quiz you can do, as well as the answers for that quiz. So with that introduction, let's get back to our introduction to the lab manual. Now, the second meeting of the semester we're going to use for laboratory training, safety training in lab, and you'll be taking a quiz on that as you are safety trained. And you have to pass that with an 80% or greater in order to attend the first real lab of the semester. So our second class meeting will be strictly safety training. If you want to review some of these laboratory rules, that'll help you understand the safety training once we get to that in class, but we will cover that in class. You'll be expected to wear personal protection equipment or PPE during this class. And we are a biosafety level 2 lab, which means that in our microbiology lab, you have to wear a full-length lab coat that's fully buttoned to avoid contamination of clothing and skin. You have to wear OSHA recommended safety goggles, splash-resistant goggles. Now, normally we provide those in lab and we disinfect them with ultraviolet radiation between every lab. However, if classes start meeting on campus during the COVID-19 outbreak, we will be expecting students to buy their own splash-resistant goggles. But again, they're not safety glasses, remember, they're splash-resistant safety goggles, and these should be available in the bookstore. Also, you'll be expected to wear gloves. We will provide the gloves. And after we're done using the gloves, they have to be disposed of in the designated biohazard container. So do not dispose of gloves in a trash can. That is an OSHA violation. And you can use your lose core points for doing that. Now let's get down to binomial nomenclature. That's kind of a prerequisite to being able to do lab 1. So as you probably remember from biology, all organisms are given 2 scientific names. Binomial means literally 2 names. And what you need to know is that there is a genus name and a species name. The first name is the genus, the second name is the species. So one of your objectives says to define genus and species. And that means that that means you should know that genus is the first of the 2 scientific names, species is the second. So in our illustrations here, Bacillus subtilis, Bacillus is the genus name, subtilis is the species name. With Escherichia coli, Escherichia is the genus name, coli is the species name. Staphylococcus aureus, Staphylococcus is the genus name, aureus is the species name. So that later on a lab quiz, if I ask you to name the genus of this bacterium, you know what I mean by genus. I want the first of the 2 scientific names. Now, when we write the scientific names of a microorganism, the person who named these organisms usually translate that descriptive name into Latin or Greek. And so we're actually writing the name in a foreign language, and English style says that when we write words from a foreign language, we italicize them. So that's why when they're written correctly, we see them italicized. In lieu of that, if you're handwriting, you would normally underline that. Now, we also have to be familiar with a little bit of the metric system, especially metric length and fluid volume that we'll be using in our class during the semester. So remember, the basic or standard unit of length in the metric system is the meter, abbreviated M, and that's a little bit longer than a yard. It's about 39.37 inches. The basic unit for fluid volume is the liter, abbreviated L, and that's about 1.06 quarts. Now, if we put a prefix in front of the basic unit, that prefix indicates either a fraction or a multiple of that unit. And there are only 4 of these prefixes, though, we need to know during the course, the ones we will use. So the prefix centa, abbreviated C, means 1/100th or 10 to the minus 2 in scientific notation. So a centimeter, abbreviated cm, would be 1/100th of a meter. And those are the larger units you see on a metric ruler, the ones that look like about ½ an inch. That would be the centimeter, which would actually be 10 millimeters, as we'll see in the next section. Now, the next smaller unit, the prefix is milli, abbreviated M, which is 1/1000th or 10 to the minus 3. So a millimeter is 1/1000th of a meter, which is actually 1/10 of a centimeter. And those are the smallest units you see on a metric ruler. Now, we're going to be using the term millimeter later on when we do antibiotic susceptibility testing, where we'll be measuring the diameter of zones of inhibition around antibiotics in millimeters using a metric ruler. And the fluid volume we'll be using is milliliter. That's 1/1000th of a liter. So again, milli means 1/1000th, so a millimeter would be 1,000th of a meter, milliliter would be 1,000th of a liter, a milligram would be 1,000th of a gram. But these two units are huge compared to microorganisms. So the most common metric unit we use is the prefix micro, which is abbreviated with the Greek letter mu, because we've already used m twice for milli and for meter, so we can't use it again. So micro stands for 1/1,000,000th or 10 to the minus 6. So a micrometer, abbreviated mu-m, is 1/1,000,000th of a meter. A microliter, abbreviated mu-L, is 1/1,000,000th of a liter. And we'll be measuring bacteria in micrometers in lab 1. And then the final prefix we need to know is nano, abbreviated N, and that's 1/1,000,000th or 10 to the minus 9. So a nanometer, abbreviated NM, would be a billionth of a meter or 10 to the minus 9 meters. And that's what we use to measure dimensions of parts of a bacterial cell or the size of a virus. So in lab 1, we're going to be measuring the diameter of bacteria or the length of bacteria or whatever in micrometers, 1/1,000,000th of a meter. And if we relate that to our system, that's about 1/25,400th of an inch. So an average bacillus-shaped bacterium, also called a rod-shaped bacterium, one that's basically kind of hot dog shaped as we see in figure 1, like this E. coli up here. Notice it looks like a little teeny tiny hot dog or like a large hyphen. That would be a single bacillus. And a single bacillus like E. coli typically is about 1/2 to 1 micrometer wide and only about 1 to 4 micrometers long. An average coccus-shaped bacterium, that's a spherical bacterium, like a single staphylococcus shown in figure 2. Now we'll see in lab 1, this is one of the arrangements of cocci called a staphylococcus arrangement, because staphyle means grapes. They look like grape-like clusters under the microscope. But if we look at just one coccus in that cluster, a single bacterium, then a single coccus typically has a diameter of about 0.5 to 1 micrometer. So bacteria are extremely tiny. A volume of 1 cubic inch could contain about 9 trillion average size bacteria. And it would take over 18 million average size cocci lined up edge to span the diameter of a dime. Or to put that into perspective in lab 1, when we see the staphylococcus in lab 1 that we see here in figure 2, note that magnified 1,000 times, that's the magnification we'll be using to observe bacteria, 1,000 magnification, that it's about the size of a period on a printed page. So when we see that, that's 1,000 times its real size. To put that into perspective, if you magnified a common housefly 1,000 times, it would have a wingspan of about 38 feet. So that's how many times we're magnifying these bacteria to look at tiny dots and tiny dashes. And of course, in a number of labs, we'll be measuring fluid volume in millimeters using pipettes. So those are the metric units we'll be using during the course, and you need to know those by the time we get to lab 1 and our first lecture. Part G deals with using a microscope. Now the actual focusing will take up in lab 1, and you'll have a couple videos on that you'll be taking pre-lab quizzes on prior to lab 1. But this is a brief description of the microscope and some of the parts. So generally, before we plug the microscope in, we turn the light intensity control dial down to 1. It should be at 1 because that's where you turn it when you put it away. But this is the light intensity dial here shown in figure 3. And that should be set at 1 before you plug it in. Once it's plugged in, then use the on-off switch on the right side to turn the microscope on. So here's the on-off switch. And when the O's pressed in, that's off. And when you push the single line in, that's on. And at that point, there'll be a faint glow coming from the microscope, but you won't see anything necessarily through the eyepieces because the light's all turned almost all the way down. Now, once you have it plugged in and you've turned it on, then adjust the voltage control dial all the way up to 6. So once it's turned on, then we can crank it up to 6, again using the light intensity control here. It takes a lot of light to see bacteria at 1,000 magnification. So we need the light all the way up when we're ready to start focusing. Then you need to adjust the eyepieces. You can pull or push them apart until you're seeing one image with 2 eyes. You need to get the distance between the 2 eyepieces or ocular lenses to match the distance between your eyes. And you need to see the whole field of view. So normally your eyes are about 3/4 of an inch to an inch away from the lens to see the whole field of view. So the ocular lenses are the eyepieces. And again, if you just grab those with 2 hands, you can rotate them up and down and increase or decrease the distance between them to match the distance between your eyes. When we put a microscope slide on the stage, we use a mechanical stage to observe bacteria. So what you want to do is to secure the slide between the 2 slide holder arms of the mechanical stage. If we look at figure 5, this shows how the slide's inserted. So when you push this knob here back, that's going to that's going to pull this arm forward so you can slide the slide in. And you want to slide the slide all the way back to the end of the slide holder so that it's held firmly in this little L shape by the edges. Then when you release the arm, that'll clamp the edge of the slide. By holding it this way then, when we move the stage with the stage controls, we can move the slide as we're looking through the microscope and move it from one place to another while we observe the slide. So that's the way the slide will be held. It's not like a toy microscope that have spring clips where you try to put the slide under the clips. The mechanical stage holds the slide from its edges like we see here. To adjust the light, we first of all use the light intensity control dial that you turned up to 6 once you turn the microscope on. So in case you forget to turn that all the way up, remember that has to go all the way up to 6, generally when we're viewing bacteria. Then we need to adjust the amount of light coming through the condenser using the iris diaphragm lever located directly under the stage in the front of the microscope. And this can be seen in figure 3A. So if you look underneath the microscope, this is the iris diaphragm lever. Don't turn this ring here, the field iris diaphragm control ring. You just leave that wide open. This is the iris diaphragm lever directly under the stage. And notice here you see 0.5, 1, 1.2, etc. So we can adjust the light. When we slide that lever to the left, that increases the amount of light coming through the lens. And when we move it to the right, that decreases the amount of light. And that's what we use to get contrast. That's what we use to improve the image. Every time we focus, we play a little bit with the iris diaphragm control lever to see if reducing or increasing the light a little bit makes the image a little more clear. And while we're looking at this diagram, these are the 2 focus or the 2 stage controls right here. So when you rotate this top one here, that makes the slide go forward and backwards on the stage. And when you rotate the bottom one, that makes the slide go left and right on the stage. And then we see our two focus controls. The large control is the coarse focus, the fine control is the fine focus. And finally, we want to check the condenser height control. There's a single knob under the stage that looks about the size of a focus knob, but it's under the stage. That's just actually used to lower the condenser to take it out for cleaning and such. But sometimes someone will grab the condenser height control thinking it's a focus knob, start lowering the condenser, and then lose all the light. Now, one of your objectives says you should be able to calculate the magnification you're using under the microscope, which is simply the product of the two lenses being used. So the eyepiece is called the ocular lens, and that magnifies the image 10 times. That's standard in the industry. We use 10x ocular lenses. The objective lenses are the lenses mounted on a turret near the stage. And as you rotate the lens, you can get different magnifications. If we look at figure 4, we see several lenses here at the turret. So these are the ocular lenses or eyepieces, and these are the objective lenses. And each one is marked with a different magnification. So if we are using our 10x objective, that's one that has a yellow stripe, and the colors are standard in the industry, in conjunction with our 10x ocular lens, then the total magnification is 10 times 10 or 100. We're seeing the image 100 times its real size. If we were to rotate the blue striped 40x objective into place, then the total magnification would be 10x of the eyepiece times 40x of the objective. We're seeing the image 400 times its real size. But almost always we're going to be using the 100x white striped oil immersion objective. So in that case, it would be 10x of the objective times 100x of the 10x of the ocular times 100x of the objective. We're seeing the image 1,000 times its real size. And that's the maximum useful magnification you can actually get with a light microscope, one that uses visible light as a light source, as opposed to an electron microscope. Now, again, as part of your pre-lab quiz for Lab 1, there's a video that covers the parts of the microscope that I just covered here. There's another video you'll be watching that tells you how to focus using the 10x objective, using low magnification. But most importantly, there's one that tells you how we'll focus using oil immersion microscopy, or 1,000 magnification. That's what we'll be using over 90% of the time in this course. So again, we'll save that for the videos, and we'll be practicing that in lab. Now, it's also important that we clean the microscope. Remember, on lenses, both the ocular lens and the objective lens, we only use lens paper. Not Kim wipes, not paper towels. They're too abrasive. They could scratch the lens, but only lens paper, which will be provided. And before you put the microscope away at the end of a lab, always remove any immersion oil from the oil immersion lens before you put the microscope away. And again, use lens paper for that. The lens paper can go in a regular trash can when you're done. Now, the reason we call this oil immersion is that we put the lens, the 100 objective lens, in a drop of oil on the slide. And here's the reason why we need the oil and why it's called oil immersion microscopy. So one of those little pesky laws in physics says that when light waves travel from one medium to another, they bend. And we can see that sometimes, for example, like in figure 6, where if we look at a pencil in a beaker of water. Now, when we're looking at the top, the light is going through glass, the beaker, and then hitting air. But in the bottom half, the light is going through glass and then hitting water. And water has a different refractive index from glass, which means the light bends differently when it goes into a different refractive index. So notice when you look at the pencil, it just doesn't look right here. That's distortion, because here we have light going from glass hitting air. Here we have light going from glass hitting water. and water has a different refractive index from glass, which causes a distortion from what you would see in air. So the same thing happens with a microscope. Now, it does happen under low magnifications. It's just that the distortion is so slight, our eyes can't detect it. They don't have a good enough resolution to detect it. But remember, when we're using 1,000 magnification, we're magnifying the image 1,000 times. We're also magnifying that distortion effect 1,000 times, and now it becomes visible to the naked eye. So the immersion oil we use is a special oil that has the same refractive index as glass. In other words, when light goes through the oil, it behaves just as if it was going through glass. And we can get that idea in figure 9 here. So if we were using a lens without oil, if that drop of oil was missing, then as the light hit the glass and then hit air, it would bend. And that bending of the light when it goes from glass into air would cause a distortion of the image. Now again, at low power, we can't detect that, but at 1,000 magnification, it makes the image blurry. So notice that the oil we use has the same refractive index as glass. The lens made of glass has a refractive index of 1.52. The oil behaves as liquid glass, same refractive index, 1.52. The slide made of glass, 1.52. But air has a refractive index as 1. And when we go from one refractive index to another, that's where the light bends and the image may bend. So what the oil does is create a homogeneous path where the light goes into glass like oil into glass. It doesn't bend, same refractive index, and that prevents the distortion of the light waves that we would see if we didn't use the oil. So it's very important that we remember to put that drop of oil on there. That is one of our foking steps. We use the immersion oil to prevent distortion at high magnification. Now, it doesn't help focus. I get that a lot on a quiz. The lens focuses. The oil prevents distortion at high magnification. And we can see that if we look at a bottle of immersion oil in figure 7. Here's a dropper bottle with a dropper in it. And this is immersion oil inside. But right now, that dropper is filled with air. There's no oil in it. And so when the light goes from glass into glass like oil into the glass dropper, but then hits air, it bends. And that bend creates the contrast we see here. So we can see the dropper clearly because it's filled with air. Air has a different refractive index from glass. But now if we were to squeeze the bulb up here and fill this dropper with oil, like we see in figure 4, it looks like the dropper disappears. It doesn't, but the oil inside now has the same refractive index as glass. So starting at the outside of the dropper bottle, all the way through the dropper bottle till you go out the backside, we have the same refractive index. Glass, oil, glass, oil, glass, oil, glass. But it's all the same refractive index, and you don't see that distortion. And so that takes care of the introduction to our lab manual. And don't forget, you do have several testable objectives under here that could appear on the first lab quiz.